Hello everyone, so in this video we're going to be covering two chapters actually. We're going to be doing 5C and 5D, and the reason for that is that 5C should be relatively simple if you've looked at my enzyme videos, because it's basically going to be mostly a repeat of that, and 5D, if you studied Area Study 1, which I hope that you did, is also just revision of that as well. So 5C and 5D are just really using what you already know and applying it to photosynthesis specifically. So in 5C, what we're doing is we're looking at the factors that can affect the rate of photosynthesis, and in turn, we're also looking at the factors that can affect the enzymes that are used in photosynthesis as well. So the first one is light. Okay, so hopefully it's quite logical to understand how light might impact photosynthesis. Light is obviously required for the light-dependent stage. So if light increases, photosynthesis will increase, and if light decreases, photosynthesis will decrease. So you'll see that in this graph here. So the rate of photosynthesis and light intensity increases. All right, makes sense until this point here. So we reach a plateau. And you'll see in a lot of these graphs that a plateau or just like a straight line, so not an increase in the rate, happens a lot. And this is because of limiting factors. So even if the light's going up, there's no point in the light going up if all of our enzymes are saturated or another input is not also going up. So like for instance, if our light is going up but we're not getting enough water, there's nothing for the light to split, so it will plateau. Okay, so as a general rule, light increase results in the increase of photosynthesis, but there's always going to be some sort of plateau point with our limiting factors. So enzyme saturation, not enough water, all of those sorts of things. So light affects C3, C4 and CAN plants all the same because they all have really similar light-dependent reactions. So we know that not much change has occurred in the light-dependent reactions. All the adaptations are in the independent reactions to combat photorespiration. So this is just an example of how different colours of light can affect the plant. So it's often found that the blue wavelength of light is absorbed best by a plant and is most, is handy for photosynthesis, and also the red. So we've got our blues and reds are the color of light that is best absorbed by the pigment chlorophyll in photosynthesis. And that makes sense because you'll see green light here is the least effective in photosynthesis. That's because the pigment chlorophyll is green. So green is going to be reflected, not absorbed in the plant. So green light is often the least effective for photosynthesis, and our blues, purples, reds is often most effective. We're going to do a prac that's going to look at this, and it will make a lot more sense then. We've then got carbon dioxide. So carbon dioxide is a really similar concept to light. So carbon dioxide is an input of the light-independent stage. So as a general rule, if carbon dioxide is increasing, the rate of photosynthesis increases. Okay, so that's a general rule. But again, we see a plateau, and this is because of limiting factors. So there's no point in having more carbon dioxide if all of the rubisco enzymes are saturated, or there's not enough light, or something like that. So all of these factors, as a general rule, will always go up, and they'll always increase the rate of photosynthesis, but these limiting factors really do always cause this plateau. If there's no limiting factors, then sure, it could just keep going up and up and up. But in real life, we know that there's normally limiting factors there. So when CO2 concentrations are low, there's also a risk of photorespiration occurring. So even though this graph, it looks simple enough, we can't forget what we've learned. So we know that photorespiration can occur, and you might see that in a graph. So even though the carbon dioxide concentrations might be rising or something like that, if the rate's not rising, it could be due to a limiting factor, but it could also be due to photorespiration occurring if it's really hot or something like that. So it's really important to keep what we know in mind when we're looking at these graphs, especially if it's a C4 or a CAN plant where it might be less affected by these things. So make sure you always keep what you know in mind. We then have water. Okay, water, same deal. So if water increases, photosynthesis could typically increase unless there's a limiting factor. So if there's not enough light to split it, or if there's not enough carbon dioxide or anything like that, then the amount of water isn't really going to help the overall rate of photosynthesis. Decreased amount of water also leads to the closing of stomata. So we know that when it's really hot and dry, our stomata will close to prevent water loss, which will in turn decrease the amount of CO2 coming in, increase O2, which can decrease the overall rate of photosynthesis because photorespiration might occur, carbon fixation might not be happening. So again, really important to note what plants you're working with. In C3 plants, this might be a real big problem, but in things like C4 and CAN plants that are not really as affected by these things because they close their stomata, etc., they might be more or less susceptible to water loss depending on the scenario. So really important to note that when you're looking at photosynthesis rates. Then we go into things that I'm hoping are really familiar to you. Because photosynthesis is heavily run by enzymes and coenzymes and all those sort of things, especially rubisco, obviously the things that affect enzymes are also going to affect photosynthesis. So temperature, pH. Temperature, all plants will have an optimal temperature at which their enzymes will work at. Anything above that will result in denaturation. Anything below that will result in freezing. Okay, so if these temperatures are not optimal, that may affect the rate of photosynthesis. Same with pH. All enzymes are going to have an optimal pH they work at. If it's more or less than that, the enzymes are going to denature, and that's going to lower the rate of photosynthesis. Enzyme inhibition as well. So we learnt about competitive and non-competitive inhibitors. So these are very much present in photosynthesis for rubisco. So remember, competitive inhibitors bind to the active site of enzymes to prevent the substrates from binding, and then non-competitive inhibitors bind to an allosteric site, which causes a change in shape, which means the substrate can no longer bind. Something that's cool to note is that many herbicides use what we know about enzymes to act on plants. So many herbicides will actually be enzyme inhibitors, and because herbicides are to prevent weeds and things like that growing, they will inhibit the enzymes that allow the weeds to grow, and that's how herbicides actually work, is by preventing those enzymes from working, which lowers photosynthesis rates, which therefore stops the plant from growing. So we've got our graph here that might look a little bit confusing, but we've got our green line, which is uninhibited. So this is the rate of photosynthesis when uninhibited. So you'll see as the substrate increases, as does photosynthesis up until this point here, which we know is due to limiting factors. So there might have enzyme saturation or things like that. We then have our competitive inhibitors. So as the substrate concentration increases and the rate of photosynthesis will also increase, again, it'll reach a certain point. And then we've got our non-competitive inhibitors. So you'll see that they're probably the most effective in stopping the rate of photosynthesis. So I would say that a good herbicide would probably incorporate the non-competitive inhibitors, because you can see that the rate of photosynthesis is really damaged by this non-competitive inhibition. So this is just a summary of pretty much everything that we've learned about in 5C, and also a bit of 5A and 5B. So it's looking at all the different factors that could influence photosynthesis, and it's also looking at increasing, decreasing, and the differences between C3, C4, and CAN plants. So this is a really cool table as like a little summary, looking at how all of these factors affect or increase and decrease photosynthesis, and also looks at the differences between plants as well. So please do pause and have a look at that and add that to your notes. Now what we're going to do is we're going to look at 5D. So 5D is really short and sweet because it's looking at things we already know. So what we're doing is we're tying it all together, and we're looking at how we can use our DNA manipulation technologies that we learnt about in Area Study 1, how we can use things like CRISPR, which is probably a trigger word for a lot of you, I'm sorry, how we can use these technologies to improve photosynthesis. So we know how photosynthesis works, but we also know that we can manipulate the DNA of plants to improve photosynthesis. And this can be really, really essential for farming practices and crop yields and all that sort of stuff that's really important for human life. So I'm sorry if this upsets you in any way. I know CRISPR is a touchy subject, but you've learnt about it, so take this as an opportunity to revise it, okay? We're not going to go through it in any detail, but I'm just going to highlight how things like CRISPR can be used to improve photosynthesis. So one way that we can apply CRISPR that we've learnt about in everyday life is to remove disadvantageous alleles or to add really desirable alleles to a plant. So an example of a desirable allele might be pest resistance, and maybe an undesirable allele might be, I don't know, they might be really susceptible to water loss or something like that. So we can use CRISPR technology to create a genetically modified organism, a GMO, a plant, corn, things like that. So we can use CRISPR to increase our photosynthesis. So think about all the things you know about photosynthesis, all of the enzymes and the processes, they're all controlled by genes. So if we can manipulate those genes using CRISPR, so either adding in a gene or knocking out a gene, we can really improve, how farmers produce crops and things like that. So like I said, so by 2050, it's predicted that agricultural productivity will need to almost double to cope with the demands of the rising global population. So obviously the things that we've been learning about are going to be really, really topical over the next few years, because we're going to be looking at ways that we can increase crop yield. So things like CRISPR, Cas9 and recombinant plasmids and all those things you learnt about are going to be really, really important. It's going to be a really growing job industry as well, which is excellent. So with factors like climate change, land exhaustion, we desperately need to increase crop yields, mostly in C3 plants. Okay, so we can do this by editing the genome. So this last slide here is just a summary of all the different things that we could do to increase photosynthesis. And this is a really good way to revise what you know, because it's looking at all the different points of photosynthesis and all the different concepts around it and how we can manipulate these things using DNA manipulation to improve them. So things like the Calvin cycle, for instance. So we know that rubisco is a main enzyme that is involved in Calvin cycle, and we know that because it's an enzyme, because it's a protein, it's coded through a gene. Okay, so we could modify the rubisco gene to make it less less able to bound to oxygen. We could modify the activity of other enzymes in the Calvin cycle. We could even turn a C3 plant into a C4 or a CAN plant just using DNA manipulation. So these are all different kinds of things that you need to be thinking about, and these are the sort of things that examiners are going to ask you to apply your knowledge to. So they're going to, obviously you need to know about photosynthesis, about rubisco, about all of those factors that influence photosynthesis, and 5D is kind of bringing all of that knowledge together to understand how we can manipulate those processes to increase photosynthesis. So having a couple of these examples up your sleeve and being able to justify them and explain why they would improve photosynthesis by using all the key terms and processes we've learnt about is going to be really, really handy for you. So hopefully that helps. If you have any questions, please bring them to class and I will see you there.